Hello everyone, myself Pillai and I am going to show my research on statistical analysis of test and reference drug using bioequivalence and population bioequivalence under the guidance of Dr. Kimia Tinani. Have you ever wondered why a generic tablet costs a fraction of the brand version, yet the pharmacist claims it to work exactly the same way? Well, every one of us know that our life depends totally on choosing the right drug. Bioequivalence isn't just about the drugs being identical in appearance, but also being identical in behavior once they enter into our bloodstream. Hence, it is a bridge between innovation and accessibility. It ensures that patients, regardless of their budget, should receive same therapeutic success, care, and safety. So, in this, we have taken our data of test and reference drug is a crossover, 2x2 crossover design with 16 subjects. Pharmacokinetic Cmax, AUC 0 to infinity, and AUC 0 to T are calculated, analyzed using log transformed ANOVA at 90% of confidence interval, and results are compared between the confidence interval of 80 to 125 range, and PB analysis indicates failure to meet overall bioequivalence due to variability. Although median responses meet the requirement, but only on the basis of median, we can't take the decision. Overall, the test drug does not demonstrate complete bioequivalence and cannot be considered fully interchangeable with the reference drug. So, in introduction, we have already seen what is bioequivalence, and it is usually conducted to compare a test drug with a reference drug to ensure the similar rate and extent of absorption of the drug in the body into the bloodstream as well, using which is estimated using pharmacokinetic parameters such as Cmax, AUCT, AUC infinity. And this is essential for approval of generic drugs as it ensures therapeutic equivalence without the need of extensive clinical trials. Traditional approaches like bioequivalence focuses on comparing mean responses using statistical methods, but they do not account for variability among subjects. To address this, population bioequivalence considers both mean difference as well as variability, which is especially important for highly variable drugs such as inhalers. And bioavailability forms the foundation of bioequivalence, describing how fast and how much drug reaches to systemic circulations. Its key parameters are Cmax, AUC, and Tmax, help to evaluate drug safety, efficacy, and absorption rate. Bioequivalence is established when 90% confidence interval, as I told earlier, the confidence interval by a regulatory guideline is 80 to 125%, and the main aim to study this study main aim of this study is to assess whether the test drug is equivalent to the reference drug in terms of both average performance as well as variability, ensuring safety, efficacy, and interchangeability. And we have taken this data of BABE from a Canada research paper, which has 2x2 crossover bioavailability studies on 16 subjects. Concentrations of test and reference were given of 0 to 16 hours, and for PB, we have taken the data from a research paper, and where the active ingredient is budesonide. Objective of the study is to demonstrate bioequivalence between the test and reference products using pharmacokinetic parameters such as AUC 02 to AUC 0 to infinity and Cmax in accordance with regulatory guideline. To evaluate bioequivalence between test and reference drug using log transform pharmacokinetic parameters with mixed effect models to evaluate intra-subject and inter-subject variability of pharmacokinetic parameter, to assess the effect of sequence and period treatment, and to compare the statistical performance of the drug reference using population bioequivalence methodology. And while looking at methodology, two sequence crossover study with 16 subjects receiving both test as well as reference drug, and blood samples were collected at multiple time points that is 0 to 16 hours. We have estimated pharmacokinetic parameters such as Cmax, Tmax, AUC 0 to T, AUC 0 to infinity, T half, which is half time, and lambda z, which is elimination rate constant. So effects considered are treatment period and treatment period, subject, and sequence, whereas AB is used to compare mean bioavailability. Bioequivalence concluded was at 90% confidence interval, that is acceptance range between 80 to 125% for Cmax and AUC. Variability assessed using intrasubject and intersubject constant variance, and high variability can be defined when it is greater than 30%. And in PB, we have evaluated both mean and variability simultaneously, which is given by mu t minus mu r square plus sigma square t minus sigma square r, which is greater than equal to theta. And if PBE criterion satisfied, we can say it is bioequivalent. Dissolution variability is assessed using span, and which is given by D90 minus D10 upon D50, where D90 is the end stage, D10 is the initial stage or first stage, and D50 can be denoted as the middle stage. Low span indicates uniform drug release, whereas high span indicates high variability. And the software used for evaluating all these pharmacokinetic parameters and analysis are MS Excel, R, and SAS. And now the question arises, why we go from PBE to BE, population bioequivalence, is especially useful for highly variable drugs. Such drugs show highly large variability sigma square in their response. Traditional bioequivalence B focuses only on the mean difference of the between test and reference products, and may incorrectly reject acceptable products when high variability is extremely high. PBE overcomes this limitation by considering both mean and variability expressed as mu t minus mu r square plus mu square t, sigma square t minus sigma square r, thus providing a more reliable assessment of drug performance across the entire population. Hence, PBE ensures consistent efficacy and safety even in the presence of high variability. So population bioequivalence decision criteria is the formula given below, which is mu t minus mu r square plus sigma square t minus sigma r square divided by sigma r naught square, which should be less than equal to theta p, where mu t and mu r are means of test and reference. Sigma square t and sigma square r are total variances. Sigma square r naught is regulatory variance, which is constant given by 0.1, and theta p is regulatory constant. Linearized criterion for testing eta is mu t minus mu r whole square plus sigma square t minus sigma square r minus theta p into sigma square r naught. And final decision is made on 95% confidence interval, where h eta should be less than equal to 0, should be greater than equal to 0, and if it is so, we can say it is bioequivalent. If it is greater than 0, it is not bioequivalent. So parameter estimates are here. D50 and span in particular size distribution, it is evaluated here as mean diameter is a particle size at 50% of, as we have earlier told, it is middle value. So 50% of particles are smaller and 50% particles are larger from the cumulative curve, which is AUC curve. And span measures the width spread of particle size distribution, which is given by the formula D90 minus D10 divided by D50, where lower span indicates more uniform particle sizes and higher span indicates wider span variation in particle sizes. Information about parameter, where Cmax is maximum drug concentration, Tmax is the time at which the concentration is at peak, AUC 0 to T is drug exposure up to up to last time. Elimination rate lambda z is elimination rate constant. Half time t half is drug half life, which is given by the formula given below. AUC 0 to infinity is total exposure. That area to infinity is where till the time end time to the lifetime the concentration of the drug into the body is what. LLQ or LLOQ or LQCT is last measurable concentration time. Overall conclusion we have drawn from this all analysis is the test formulation cannot be considered bioequivalent to the reference formulation as per regulatory guideline, and the test and reference formulation do not meet the overall bioequivalence requirements when performed under log transform parameters using mixed model. Overall, the study shows that the Cmax has high variability, which means that peak drug concentration changes a lot within the same subject. For AUC 0 to T and AUC 0 to infinity, the variability is moderate, which shows better consistency between test and reference. Between different subjects, the variability is also moderate for all parameters. So overall, there is moderate to high variability, especially for Cmax. ANOVA analysis shows that sequence, period, and treatment effects were not statistically significant, that is, P is greater than 0.05, indicating no carryover effect, no period influence, and no significant difference between test and reference formulation. Thus, crossover design is valid and appropriate for bioequivalence assessment. Although the test and reference drugs show similar median response D50, they differ significantly in terms of overall performance and variability, that is span. Since population bioequivalence considers both mean and variability, the failure in variability leads to the conclusion that the formulation are not population bioequivalent. The statistical softwares used are Excel, R, and SAS. Thank you.